Good morning, afternoon, or evening, wherever you may be. Welcome to the October edition of AJP-Heart and Circulatory Physiology video table of contents, or from the editor's desk. From now on, I will be making these videos once per month, given that the APS will be publishing articles as they are ready. So each video will be a little longer than usual, and I apologize for that. This month, we have published 2 reviews, 25 original research articles, 2 editorial focus pieces, and 1 letter to the editor. So let's get started. We have a lot to read. In a review by Draeger et al., issues related to the association of obstructive sleep apnea and cardiovascular disease are discussed. The paper reviews animal data from experiments targeted to uncover the mechanisms of this association. This paper is part of a collection related to last year's Pan American Congress of Physiological Sciences. Lucidi et al. Studied the role of chloride intracellular protein 4, CLIC4, in cerebrovascular collateral vessel development. Deletion of this protein had little effect on embryonic collaterals. However, during maturation, excessive pruning of collaterals developed. In adults with CLIC4 deletion, a larger infarct in response to middle cerebral artery occlusion was observed, along with reduced VEGF expression. This protein appears to be critically important for the development of cerebral collaterals in the mature animal. Gong et al. Showed that electron transport chain coupled mitochondrial flash is a novel biomarker for monitoring mitochondrial respiration in cultured adult cardiac myocytes and in the perfused beating heart. Confocal imaging of mitochondria was observed in a preparation using mitochondrial circularly permuted yellow fluorescent protein. They found a close coupling between flash frequency and the electron transport chain electron flow as measured by oxygen consumption rate. These results suggest that monitoring mitochondrial electron flow by real-time imaging can be used as a novel marker under various physiological and pathophysiological conditions. Beaumont et al. Determined whether chronic vagal nerve stimulation alters myocardial infarction-induced remodeling of intrinsic cardiac ganglion cells and cardiac tissue. Guinea pigs were subjected to vagal nerve stimulation following the creation of an MI. Voltage neurograms were made from whole mount cardiac ganglia 90 days post-MI. In general, vagal nerve stimulation reduced ganglionic neuronal excitability and reduced pro-apoptotic markers in myocytes. This study shows that vagal nerve stimulation is efficacious in preserving myocardial function following MI by both neural and myocyte mechanisms. In another outstanding review, Leone et al. Discussed the methods currently used to evaluate myocyte proliferation. A review of the literature using both zebrafish and mammalian models is discussed, including man. You'll enjoy this concise review. Finally, D'Souza et al. Investigated the role of ACE on cardiac fibroblast physiology. Adult male spontaneously hypertensive rats were treated with vehicle or the ACE inhibitor enalapril for 2 weeks, followed by a 2-week washout period and a subsequent 7-day challenge with the NOS inhibitor L-NAME. At the end of the study period, cardiac fibroblasts were isolated from control and enalapril-treated left ventricles to assess proliferation rate, collagen expression, and chemokine release in vitro. Enalapril reduced proliferation of fibroblasts and collagen expression. These findings demonstrate that in vivo L-NAME treatment produced phenotypic changes in fibroblasts that persist in vitro. Overall, this is the first demonstration that transient ACE inhibition can produce a persistent modification of cardiac fibroblast phenotype to one that is less inflammatory and fibrogenic. Thank you for watching this edition of AJP Heart and Circulatory Physiology video table of contents for October. As always, please let us know if you enjoy these short videos and follow us on Facebook and Twitter. Of course, don't forget to send us your best work now, and I'll see you next month.